guys, here we are. We're looking at one of my favorite chapters. This is chapter 3, the molecules of cells. And this is a really interesting chapter. And because I'm doing this as a video lecture, I'll probably get to give you guys a lot more information or be able to explain a little bit more than I maybe would have had time to do so in lecture. So good thing. By looking at the first thing to introduce this chapter is really kind of a mind-blowing idea, and I want you to really kind of think about what this means. Lactose intolerance. This is something that's, some of you may be lactose intolerant. You may know someone, maybe your parents are lactose intolerant. It's one of those things where lactose intolerance is something that you develop as you get older. Very rarely do you find a child that's lactose intolerant. So let's think about why this is the case. Milk. Milk is not something that you as an adult should be drinking. There's no need for it. A lot of people, you may hear commercials and advertisement that say, hey, you have to get all your calcium from milk. That is just simply not true. There's so many other sources for calcium that milk is not a necessity in your life. Think about where milk comes from. Let's think about why we have milk and where it comes from. Milk comes from the breast of a mother, whether it be a cow or a human being that's nursing a child. It's meant to nurse a child to, a certain point in life. We as humans have started to consume cow's milk to make cheeses and other things. So the problem lies in the way your body is made. Lactose intolerance comes from the fact that you're not supposed to be drinking milk. Therefore, as you get older, your body is designed to make an enzyme called lactase. Okay? Lactase is what actually breaks down the lactose in the milk. So babies have lots of lactase enzyme because they're needing to break down milk. Well, as you get older, you're not supposed to be drinking the milk. So therefore, evolutionarily speaking, you're not supposed to be consuming the milk. So why would you need to make the enzyme? So what happens is as people get older, they stop producing lactase because they're not supposed to be having the milk. So because they're not producing lactase, they no longer can break down the lactose. So that's where they develop lactose intolerance. So that's the introduction. Looking at the role of the enzyme lactase with lactose. Again, the enzyme lactase is going to be what breaks down lactose. And just to kind of further give you an idea here, when you're looking at enzymes, we'll look a lot at enzymes. We know something is an enzyme by the ending ase. It'll be something ase, like this one here is. So lactase is going to break down lactose. With that being said, sugar is going to have the ending ose, O-S-E. I don't know why that O looks funny, but that is an O, O-S-E. All right, so that's the introduction here. But I hope you kind of take something from it because I think it's really interesting. And something else to think about here, and I think I always like to have everybody think about it. We are one of the only species, so species being we're Homo sapiens. We are Homo sapiens. We are the only species that will give another species milk to our children. Things that make you think, right? We give our kids cow's milk, sometimes goat's milk. All right, cows make milk for cow babies, okay? Goats make milk for their babies, right? And it's interesting how we'll give another species milk to our babies. Isn't that crazy? Because you're not really given the opportunity. I doubt a cow would give its baby another species milk. I mean, it just really doesn't happen. In nature, you don't really see other species nurturing other babies with, like you don't see a cow necessarily nursing a goat's baby. Like it just doesn't really happen. I just think it's interesting. So again, something to think about. That's the introduction. We're looking here at lactose and lactose intolerance. So that's kind of the introduction to this chapter, and we're going to move on. So big ideas from chapter 3. The first thing we're going to talk about is organic compounds. Like, what is an organic compound? That sort of thing. Then we're going to introduce carbohydrates. What's a carbohydrate? What are the properties of a carbohydrate? Lipids, what are a lipid? What is a lipid, and what are the properties of lipids? What's protein? What makes up a protein? What are the properties of a protein? All this other stuff. And then same thing with nucleic acids. We're going to break down each of these guys. When we're looking here, we're going to be studying macromolecules. All right, so macromolecules. Let me write this word down. Macromolecules. Carbohydrates, lipids, proteins, and nucleic acids are all macromolecules. Macro just meaning large molecules. So all of these guys are really large, and basically, so what this chapter 3 is ultimately looking at is the study of macromolecules. All right, so introduction to organic compound. Here, we're really going to look at how fabulous carbon is. So that's the goal. You need to be building on the knowledge that we learned from chapter 2, the chemistry section. So make sure that you've gone through all of chemistry and you really understand the bonding before we jump into this one. So introduction to organic compounds. All right, almost all molecules of a cell are made up by carbon of some sort. So carbon bonded to other carbons, or carbon bonded to other atoms or elements. Carbon-based molecules are called organic compounds. Okay. By sharing electrons, remember carbon's going to share. What type of bonds are these? When they're sharing electrons, remember these are covalent bonds. Sharing electrons. Carbon's going to always make 4 bonds, which the fact that it can make 4 bonds allows carbon to be branchy if necessary. One of the simplest carbon molecules is methane. Remember, we talked about this in the chemistry section. It's the simplest organic compound. It's made up of 4 covalent bonds with 4 hydrogen molecules. Each of the 4 lines in the formula of methane represents a pair of shared electrons. Let's go back for just a second and do a small, tiny chemistry review. I want to look at here. I gotta get my pen back. So just a small review. Valence electrons. Remember, these are the ones involved in bonding. I just copied and pasted and put this into a slide. Remember, we're ignoring all of these because these are transition metals. Okay, so we're not really concerned with them for this class. When we're looking in this one, everything in this column has 1 valence electron. Everything in this one has 2 valence electrons. Everything in this one has 3. These have 4, 5, 6, 7, and 8. So here's my carbon, right? Carbon's going to have 4 valence electrons. Now, think about what that means for a second. Let's go to the next slide. Remember, valence electrons are the electrons involved in bonding. So carbon does in fact have 6 total electrons. It just has 4 valence electrons, and those are the ones that we care about. So what does that mean? So get my pen back here. So we saw that it looked like this. It was like 6, carbon, and 12. So a mini review. Let's look real quick. So my protons, my neutrons, and my electrons. Let's calculate those for a second. Remember, protons come from your atomic number, which is 6. And a neutral atom, which is one we're kind of looking at on the periodic table, protons and electrons are the same. And then the neutrons are going to be 6. So if we were to draw this guy out, the atomic structure, we would say protons have 6, neutrons have 6. We do a little circle. Our first electron shell here can hold a maximum of 2 electrons, so we're going to draw those here. There's 1, 2. And our next energy level, or electron shell here, can hold a maximum of 8. So We have 6 electrons total. We've already placed 2. That means we have 4 left. So valence electrons are on the outer energy level. Energy level. Okay. So we said carbon has 4 valence electrons. So remember when we did that Lewis dot diagram, we said treat this, so when we're doing the kind of like the abbreviation and drawing the structures, we said carbon, imagine carbon has 4 sides, 1, 2, 3, 4. And when you're going to place the electrons, let's do the electrons in a different color, see if I can get that. Let's see, color, let's do them in blue. We have, remember you only place around carbon for the Lewis dot structure, so let's type in Lewis dot. I don't know why it's so shaky looking. All right, so here we go. We have only map out the valence electrons, so valence electrons here. So we're going to say 1, 2, 3, 4. Remember, each side gets an electron before you double up, so you can't say 2 and then 4 like that. Now let's look at hydrogen. Remember, hydrogen just has that 1. Hydrogen just has 1 electron, so it only can look like this. It's only got that 1 electron. So when you're making something like methane, it's going to be carbon with its 4. And then hydrogen. That just means that there's 4 hydrogens and 1 carbon. It's going to look like this. All right, and here, let's do a different color for the, let's do green. Green, we're going to cut right here. These 2 are shared. This green bond here is a covalent bond. Covalent bond, and that means sharing. That is supposed to be an R, but it looks more like, I have no idea what that is. Sharing 2 electrons. You all on the same page? Now here's the thing about carbon. I'm going to back up and clear this off here. So this is kind of our review right now of carbon. We're saying that carbon can make 4 bonds. It really makes carbon special because it can do this. And what I mean by that is, we got a clear screen now. When you're looking at carbon, carbon can make long rings, meaning I could say, let's change that color. Let's see, red. Carbon can say, I could just go for days, right? Let's just make it pretty long like this. Methane, ethane. There should be 9. This is a no-name carbon. So actually, let's make it smaller just because I don't want it to get too overly complicated. Let's do 5. Let's do 4. 1, 2, 3, 4. And imagine I really can make this really, really long, but we're just going to try to keep it simple, but to make it effective. So here we have 4 carbons, all right? So it would be the molecule that I'm going to draw out. It's going to be C4. Now, here's the thing when you're looking at these. You need to have 4 bonds all around. Normally when we're looking at organic compounds, they're mostly going to be made up of carbon and hydrogen. So we'll say 1, 3, 4. So around this carbon, we have 1, 2, 3, 4. Around this carbon, we have to have 4 as well. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. All right. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, H10. This is butane. All right. Another example here would be to look at 1, 2, 3, 4. 1, 2, 3. Notice around every 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. Let's count this one. There's 4 carbons. And there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here's the thing about this one. This one has this branchy side. Notice it's still got 4 carbons and it has 10 hydrogens. This is called isobutane. Oh, I'm making tane. Okay. Now here's the thing about these. These guys are called structural isomers. They have the same chemical formula, but they are put together different. This is the start of it all, guys. The big picture right now is what we're really going to kind of get at. If the structure is different, the function is different. These guys have the same chemical formula, but structurally, they look very different. If they look different, they're going to act different. That is a really, really big deal. But to get an idea, this is things that carbon can do. So there's some more examples, and I'll throughout this presentation. This is methane again. If I were to have a stick model to build methane, this is methane, CH4. So it's the same thing that we were looking at here. But when you put it together with sticks, it's called a tetrahedron because it takes this tetrahedral type shape where it has 4 points. Methane and other carbons that are composed only of carbon and hydrogen are called hydrocarbons. Carbon attached to lots and lots of hydrogens. So kind of giving you an example that we looked at a minute ago with butane or isobutane, those are all considered hydrocarbons. Carbon can make straight chains, it can make branches. So when I look here, example here, like when I did butane, branchiness would be isobutane. All right, because it had that branch off to the side. Or here's another thing. Carbon has the ability to make these rings, which is interesting. So I'm going to show you the structure of the ring. So compounds with the same formula but different chemical arrangements are called isomers. Again, example would have been butane and isobutane are examples here.
Isomers are compounds that have the same molecular formulas but different three-dimensional structures. The difference between isomers can literally mean the difference between life and death. L-DOPA, shown on the left, reduces the symptoms of Parkinson's disease. Its isomer R-DOPA on the right has no effect on the disease.
Structure follows function. Lock it in your brain. If it's a different structure, it's going to have a completely different function, all right? Very, very, very fundamental aspect. You're going to hear this. It's a fundamental aspect along all levels of biology. If you take Bio 102, we look at anatomy and physiology. You look at the anatomy to figure out the physiology, like what's it going to do based on how it looks. So it's really, really, really important. Here's looking at some of the carbon skeletons.
The carbon backbones of organic molecules may vary in length and in location of single and double bonds. Molecules may be straight or branched and may even contain rings of atoms.
So there's that carbon ring. We're going to explore more. Honestly, especially in this chapter, when we're looking at macromolecules, we look at a lot of rings. So this is an important idea. Here's looking at the different types of isomers. The ones I showed you with butane and isobutane are called structural isomers because they have that, they're kind of put together differently. There are some others, and let's take a look at those.
3 types of isomers are structural isomers, which differ in the covalent partnerships between their atoms, geometric isomers, which vary in arrangement of atoms around a double bond, enantiomers or stereoisomers, which are molecules that are mirror images of each other, like left and right hands.
All right, so here's some examples. So here when you're looking at length, notice that by just adding one more carbon here, you just continue to make this chain longer, and you could really just keep going and going. Here is another example, 1 butane versus 2 butane. Now you're probably thinking, well, what's that mean, the 1 and the 2? What that does is give you an idea of, if I were just to say 1 butane or 2 butane, what does that mean? That is to tell you the location of the double bond. This is on the first carbon here. This double bond is located on the second carbon. That's where they get the 1 and 2. They're still the same. They're isomers because if you count the carbons and you count the hydrogens, they're the same. You're just changing the location of that double bond. Branching, like I showed you earlier with butane versus isobutane. Here, this is probably a better looking picture because it just looks a lot neater. But you have this branch, this is branched off here. All right, and then let's look at these guys here. Cyclohexane versus benzene. One thing I want to point out, not really that important for this class, but something to kind of lock in the back of your brain. When you have all single bonds, you're going to have the ending -ane. Ending is going to be -ane. If you have at least one double bond, the ending is going to be -ene. So notice you have a double bond, you have the -ene ending. All of these have all single bonds, so the ending is -ane. Okay? All single bonds, all single bonds. So just to kind of clarify, this is a single bond when it's just one line like that. So that is equal to a single bond. And if it looks like that, I should probably make this into an arrow so it doesn't get confusing. And if it looks like that, it's a double bond. This guy here is all single bonds, but notice this one has 3 double bonds, right? So that's a benzene. Benzene has 6 carbons. Cyclohexane has 6 carbons. What's changing here is the number of hydrogens. You're going to have more hydrogens in your cyclohexane versus your benzene. All right, so looking here, unique properties of an organic compound depend on the size and shape and the groups of atoms that are attached to the skeleton, like the main component of, they're the main aspect of the molecule. Kind of going along with what I said before, structure follows function. If it has a different structure, it's going to have a different function. What we want to look at here is comparing 2 things. Sex hormones, testosterone and estradiol. Estradiol, kind of think of that as a more concentrated form of estrogen. They differ only in the groups of atoms highlighted in figure 3.2. So let's look. Testosterone, estradiol. So what are we looking at here? First, now I want you to understand something. We kind of jumped right into this notation. At every one of these, all these points here, these are carbons, right? Carbon, 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 okay? This is a double bond. So every one of these points is a carbon. This is a hexane, right? Because it's got the 3 double bonds. It's 1 carbon, 2, 3, 4, 5, 6. So notice here that you have 3 double bonds. This one has that double bond with an oxygen. And then you have a methane group. Or sorry, not methane, sorry, methyl group. And you have these parts are all the same. All this is the same. This aspect is the same, okay? So similar structure, so they may be somewhat alike, okay, in which this case they're both steroids, but they're function is completely different. Estrogen is going to function in the body completely different than testosterone does. Structure follows function. A different structure, a different function. All right, so just because they're similar doesn't mean they're the same. Okay, and this is the same thing again. All right, these highlighted groups, the oxygen here, this green group here, this pink group here, these guys all have names. All right, we call those functional groups. The reason we call those functional groups is because they have general behavior. So they all kind of function in this very similar way. And I'm going to come back to this word in just a minute. I'm going to look at the bigger one. If it's this pink group here, this is called the hydroxyl group. These are going to form things like alcohols. So it's going to have that OH. The carbonyl group, we'll find in aldehydes and ketones, will be carbon double bonded to an oxygen. Now, if it's on the end of a structure, then it's an aldehyde. If it's in the middle, it's a ketone. So location, location, location in this aspect. Again, if it's on the end, it's an aldehyde. If it's in the middle, it's a ketone. Carboxyl is an important one here. You need to make sure that you know this one. Know this one here, definitely. All right? Carboxyl. Carboxylic acid. Notice it's COOH. You have a carbon double bonded to an oxygen, and then you form an OH group here. It's almost like taking these two and putting them together. So you have the OH group with the carbon double bonded to the oxygen. So this is called a carboxylic acid. Whenever you lose a hydrogen here, it's called ionized because you've turned this into an ion. All right, no, for the sake of this class, just knowing this part is fine. All right, let's look at the rest of the functional groups. The amino group, I gotta go back for just a second. This is another one that you should definitely know. All right, you need to know amino. That's important. Again, amino is NH2, so this is an amine group. If it's ionized, you turn nitrogen into an ion by adding an extra hydrogen. This is fine here. Just know the amine group. The next one you should know is the phosphate group. Now here's the thing. For those of you who are used to chemistry, this is something that's a bit of an adjustment even for me. I love my chemistry. We do something bad in biology. There's a lot of business going on here. There's a lot of things happening in a phosphate group. But we love to write this as phosphate in chemistry. In biology. So when you see P with a little I down here, it's an inorganic phosphate. It is not phosphorus, even though P by itself in a normal situation would be phosphorus. But in bio, when we write this, we are talking about a phosphate group. So it can be extremely confusing. So just kind of know this, and I'll kind of remind you as we're going through. But when you're learning, it's nice. They kind of do this throughout the book, but they never really explained that P by itself is in fact a phosphate group and not a phosphorus. Phosphate group. Phosphate group, this is phosphate. Actually, this is 3 phosphates, so it's not really that great of an example. What you're looking at here is P. This is 1 phosphate. 1, 2. All right, this is it right here. 1, 2. I think this one is charged too. 1, 2, 3. This one is typically charged as well. So this one's going to bond to something else. So this one's not charged. So these two are charged. So you have it bonded onto something here. So it'll be attached to something, and then it'll look like that. This is 1 phosphate group. What this is, this molecule right here that's got 1, 2, 3 phosphates, this is ATP, adenosine triphosphate. Tri because it's got 3 of them. This is a very important guy that we will talk about through the course of this class, especially when we're talking about photosynthesis and cellular respiration. This is the energy currency for our cells. Our cells store energy in the form of ATP. This guy here is a bundle of energy. And let's think about why. This guy is negatively charged, this one's negatively charged, this one's negatively charged, this guy here is negatively charged. Think about what this means. Have you ever tried to take two magnets that are both negatively charged and put them together? You probably have. It's fun to do. They repel each other, right? They do not. I mean, think about the amount of force you have to put behind those two magnets to get them to come close together. It takes a lot of force, right? That's the same thing that's happening here. These guys are not sticking together because they are doing it willingly. A lot of energy and a lot of force had to go into putting these phosphate groups beside one another. That's built up energy, okay? It's built up resistance, if you want to think about it like that. So when you pluck it off, so if I were to take one of these phosphate groups and take it off, pluck it off, imagine the relief this guy's going to have because he's no longer being forced next to that other negatively charged molecule. So this is an energy storage molecule. This is a lot of built up energy because they're all negatively charged and they're not happy with being bonded to one another, okay? That's the idea behind this. That's a whole nother lecture and everything else, but just to kind of give you an idea of why this one's important. But please be familiar with phosphate group and an amino group, and then the carbonyl, what other one did I give you? The carboxyl group from the previous slide. I want to go back for just a minute to this word here. Hydrophilic. Okay. These functional groups are all polar. Some compounds contain them are typically hydrophilic, water-loving. So all polar, hydrophilic. We said water is polar. So anything that is also polar is hydrophilic, like dissolves. That didn't look right for some reason. Like dissolves like. Polar things like other non-polar. Polar things like other polar things, all right? Just like non-polar things will like other non-polar things. If something is hydrophilic, I'm going to just use this space here at the bottom. It's not relevant to this slide. But actually, let's get back for just a second. Let me write the word here because that makes more sense. Pen. There's also hydrophobic. These are going to be water-fearing. Okay, water-fearing, meaning anything that's hydrophobic is going to be typically nonpolar. All right, nonpolar things want nothing to do with a polar molecule. They're not the same. They're not going to intermingle. All right, so here are the functional groups, hydroxyl group, carbonyl group, carboxyl group, amino group, phosphate group. All right, so getting into the macromolecules. So what we're going to do here is we'll break down each one of these, starting with carbohydrates. But just giving you an idea, here are the four classes of molecules that are important to us. Carbohydrates, lipids, proteins, and nucleic acids. All right. So first things first, macromolecules. We're calling them macromolecules because they're large, all right? Another word, and here it's going to kind of be a little bit confusing because we're going to start, it's like welcome to biology. The thing we like to do in biology is we like to give you a lot of different words that all mean the same thing, okay? Once you kind of get that idea that we're going to do that to you, it makes it a little bit easier. And I'll try to help with putting the words into groups so that you can understand them. I apologize, but I didn't make it up. So what we're going to do here, macromolecules and polymer really kind of mean the same thing. And here's what I mean by that. Macromolecules are polymers, okay? Macromolecules are essentially polymers. Polymers are made up by monomers. And here's what I mean by that. So think about a little train, okay? So like a boxcar train or something like that. The entire train is large, right? So it could be called a macro train, like a large train. But we could also call it a polymer, all right? The reason why is because it's made up of several monomers. The monomer of that train would be the boxcar itself, All right, so it takes several boxcars to make up the entire train, which is a polymer. And it's a macromolecule because it's so large. We're going to build on this idea. Just kind of keep it in the back of your mind. I'm going to keep using these words. You got to get used to the vocabulary here. All right. In order to put monomers together, we're going to do something called a dehydration reaction. All right. When you're thinking about these words, think about what that means. When you're dehydrated, you've lost your water, right? You do not nearly have enough water in your body. You're dehydrated. You've lost water. By definition, a dehydration reaction is going to remove water. Okay? If you want to break apart a polymer, you put it through something, and I'm going to say the word wrong in order to emphasize something to you. It's called hydrolysis. I say hydrolysis. Hydrolysis. We looked at this word lysis before. Hydro means water. But lysis, remember we looked at that when we talked about the chemistry section. That is to break. So by definition, this word physically means to break using water. All right? So if you want to break a polymer apart, you put it in water. All right? These reactions, both hydrolysis and dehydration synthesis, are mediated by enzymes. That means you have to have an enzyme for that. Enzymes are specialized macromolecules that speed up chemical reactions. All right? We are going to do an entire lab dedicated to enzymes, which I think will be really helpful. You guys do a report and everything else. Enzymes are really important. They have, you use specific enzymes for a specific task. Remember we talked about, what am I trying to spell here? Lactase. Lactase was the enzyme we talked about at the very beginning. Remember, and lactase breaks down, or is important in the breakdown of lactose. All right, so that's an enzyme that's needed to break down lactose. That looks like the same word. That is in fact an O and not an A. Let's fix it. Oh, there it is. Okay, next slide. So hopefully let's take a look at this for a second.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
Notice she used the word hydrolase. Okay, one thing you'll kind of notice, the name of the enzyme will have something to do with the function of it. So it is a helpful tool to use. All right. So a cell makes a large number of polymers from a small group of monomers. For example, proteins are made up of only 20 different amino acids. DNA is built from just 4 kinds of monomers called nucleotides. The monomers used to make up polymers are essential to the universe. So what we're going to do is, whether it's a carbohydrate, whether it's a lipid doesn't really have monomers, but that's a whole other thing. So whether you're looking at a carbohydrate, a protein, or whether you're looking at DNA, all of these guys are made up of monomers. So whenever you, at the end of this, you should be able to say, what is the macromolecule and what is the monomer here? That's the goal. So, and I'll make you a table as we're going through this. So here is just kind of showing you if this is a short polymer here, this would be some sort of sugar or something. if you're putting these guys together, you're going to lose water. If you lose water, it's by definition a dehydration reaction to form this. So these guys come together, you lose it. If I wanted to break this guy, I would add in water, and it goes back to how it was at the beginning. These guys are, this puts dehydration reaction, puts them together. Hydrolysis takes them apart. All right, carbohydrates. This is the first macromolecule that we'll discuss. So we're going to learn all about carbohydrates and why they're important. And before we get started with the carbohydrate, I want to give you a definition for a carbohydrate. I'm not going to give you some long, blown out definition. I want you to be able to look at these words and break them down without mindlessly memorizing some definition, okay? Carbohydrate. When you're seeing this word right here, carbo, hopefully you're thinking carbon. And then hydrates, I want you to, what hydrates you? So this is carbon. What hydrates us is water, right? So water is made up of hydrogen and oxygen. Or if you can kind of think of it as like H and O or H2O, whatever. Anyway, in any case, in any way, shape, or form, it's going to be hydrogen and oxygen. So a carbohydrate is a molecule made up of carbon, hydrogen, and oxygen. By definition, that is the definition. Okay? Carbohydrates range from small sugar molecules, called monomers, to large polysaccharides. Sugar monomers are called monosaccharides, such as those found in fructose, glucose, and honey. Here's where we're going to start using some words interchangeably. Sugar. Anytime you see this word saccharide, think sugar. A monosaccharide just means one sugar. This means many sugar, okay? Our carbohydrate is made up of many sugar molecules, all right? Now, think about this, sugar. It's the beginning of the year. People are making New Year's resolutions, and a lot of people want to lose weight to start the new year off. A lot of times, the most common things is to do fad diets and things like that. One fad diet that seems to kind of go and come, but most of you have probably heard of it, is the Atkins diet, or they call it, it used to, I don't know if they still call it the Atkins diet. It used to be the Atkins diet, but it's called the no carb diet or low carb diet. The reason people want to get their carbohydrates down is because of this old sugar. Sugar, sugar, sugar. Now, we could do an entire lecture on sugar, and I may, if I have time, talk some more about the role of sugar in the body and how the brain processes sugar and all this other fun stuff. But here we're just looking at, we're going to look at how carbohydrates are made up of sugar. So sugar monomers, so kind of think about monomers as just like one piece. That could be fructose, glucose, just one of them though. A whole carbohydrate could be like 10 glucose molecules or 10 fructose molecules, or it could be 2 fructose with 5 glucose all together to make one carbohydrate molecule. All right, so it's going to be several of these. So here's honey. Monosaccharides can be hooked together by dehydration synthesis. So if I were to take 2 glucose molecules and put them together, I need to do a dehydration synthesis like we were looking at earlier. If I put lots of them together, I make a more complex sugar, and a more complex sugar is called a polysaccharide. The carbon skeleton of monosaccharides vary in length. Glucose and fructose are 6 carbon sugars. Here's some words I want to introduce to you. Glucose and fructose are 6 carbon sugars. So 6 carbon sugars, they're hectose. That's what they're called, hectose sugars. Others could have 3 to 7 carbon atoms. Monosaccharides are the main fuel of cellular work and used as raw material to manufacture other organic molecules. So here is glucose versus fructose. Now let's get back to what we were looking at earlier. We have 6 carbons, right? This is an aldehyde. This is a ketone. All right? Structure follows function. They're similar in structure because they both have 6 carbons. They're going to have the same number of oxygens, the same number of hydrogens. But structurally, they're put together differently. This guy here is considered an aldehyde. This one's considered a ketone. The functional group has moved. If it's a different structure, it's a different function. Your body will respond differently to glucose than it will to fructose. Many monosaccharides form rings. The ring diagram may be used as an abbreviated form. When we use it, and I'll show you my lazy man abbreviation for ring diagrams, it will not show all the carbons. So it's kind of going back to when we were looking at the testosterone and estradiol earlier. Do you know when I was going in and adding in the carbons? That's a shorthand because the picture gets overwhelming when you have all the carbons in there, so they just leave a dot to show you that. So here we're looking at, structural formula looks like this. So it's kind of showing you all the carbons. This is an abbreviated form. And then this is a simplified structure. Now here, one thing I want to point out is the way this is numbered. There's 1, 2, 3, 4, 5, 6. You always label clockwise, all right? So this is the first carbon, the second carbon, third carbon. This is important because we'll look at this later. This is an abbreviated structure. And then the simplified structure. Sometimes with the simplified structure, you may see something that looks like this, because technically there's a carbon here and there's a carbon here that they're not really showing. So if I were to draw this guy out, I'd probably draw him more like this, because at this point, you would only think that there's 1, 2, 3, 4, 5. Now there's the 6. So this is a more accurate simplified structure. All right, so 2 monosaccharides, or 2 monomers, can be bonded to form a disaccharide. So imagine if I wanted to do, glucose plus a glucose. That's going to be a disaccharide. The disaccharide sucrose is formed by combining glucose monomer and a fructose monomer. So glucose plus fructose equals sucrose. So this is a monosaccharide plus a monosaccharide equals a disaccharide. So I'm going to write mono plus mono equals di. Got it? So the disaccharide maltose is formed from 2 glucose molecules. So disaccharide maltose is formed with 2 glucose molecules. This will be important when I want you to think about this whenever we're doing the lab for this particular chapter. Let's take a look at this video.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of 2 linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Remember, and they were showing you guys this, when you make this bond here, you're doing that through dehydration synthesis. Okay, all these are put together by dehydration synthesis. So here's that dehydration synthesis to make maltose. So glucose plus glucose, again, dehydration synthesis. Let's write that on here. or dehydration reaction. I like to think dehydration synthesis because you're putting two things together. It's a reaction here, so dehydration reaction or dehydration synthesis means the same thing. All right, so let's take a second and talk about the connection here. What is high fructose corn syrup, and is it the blame for obesity? Quite frankly, the question's still out. There a lot of research to support that high fructose corn syrup is the cause for obesity, and we're going to go through some things here looking at it. But what is it? Most sodas, fruit drinks contain high fructose corn syrup. Lots of things have them. Cookies. I mean, you should just kind of go through the store, go through your pantry, look at the back and see how much has high fructose corn syrup in it. Fructose is sweeter than glucose. Well, what is fructose? Fructose looks like the word fruit, right? Fruit is naturally sweet. If you want something sweet, have it from its natural source, fruit. Fructose is really, really, really sweet. It's sweet. We think it's sweeter. The natural source is fruit. It's much sweeter than glucose. What happens here to make high fructose corn syrup? Glucose atoms are rearranged to make glucose the isomer fructose. So they add things into regular glucose. So they extract glucose from corn and they add chemicals to it to make it fructose. So this isn't a natural product. It's extremely concentrated. It is cheap. It's really, really, really cheap. It's easy to get to dissolve in things. Like it's easy to put into drinks, cakes, cookies. It makes it sweet. It's really, bottom line, it's cheap. If you're, let's say you're a company that's making cookies. If you can go and buy, let's say you need 100 pounds of sugar a year, which is a lot of sugar, that could cost you several $1,000s. But what if you could get the same amount of sugar for your recipe, but for much less because you use high fructose corn syrup? It's all about the bottom line. I could use high fructose corn syrup. It's much sweeter than my table sugar, which is sucrose. And I could use less of it, and I could save my company a lot of money. That's the bottom line here. High fructose corn syrup is used to sweeten many beverages. It consists 55% fructose and 45% glucose. It's a mixture with about the same proportions as sucrose. Most scientific studies have not shown health consequences associated with replacing sucrose with high fructose corn syrup. Sucrose here is table sugar. This book was released in, let's see, your book was published in 2015. So I do have some things I want to show you about this. According to, although high fructose corn syrup consumptions have declined somewhat in recent years, obesity rates continue to rise, with almost 36% of United States adults now considered obese. Good health is promoted by a diverse diet with proteins, fats, vitamins, minerals, complex carbohydrates, and exercise. Now, they're giving you an example here of, Coca-Cola, and the second ingredient is high fructose corn syrup. So lots and lots of sugar. Well, what does this really mean? Okay, so I just want to kind of introduce some other things. Number one is this journal here. This was published in 2010, and it says high fructose corn syrup causes characteristics of obesity in rats, increased body weight, body fat, and triglyceride levels. What they're showing, what this research paper shows, and this was published in 2010, these rats maintained a diet rich in high fructose corn syrup for 6 to 7 months and showed abnormal weight gain. What they've noticed, and then something that kind of collaborates, kind of goes along with this, is kind of the big picture. These rats do experience weight gain, abnormal weight gain. There's a lot to be said, not just about high fructose corn syrup, but just about sugar in general. Your body needs sugar for energy. But does it really need this high fructose corn syrup? This is not a natural sugar. So think about it. When you're exposing your body to something that's not natural, you have a risk of, your body not processing it the way you would think to process it. Another thing, I've seen studies that show that high fructose corn syrup, when it's processed, has levels of mercury in it, which is something that we all know that mercury is a heavy metal that's not good you don't want to be consuming that. So just some things I want to kind of show you I think is interesting. Is a commercial here. Let's see if I'm not going to make it big.
Wow. You don't care what the kids eat, huh? Excuse me? That has high fructose corn syrup in it.
And?
You know what they say about it?
Like what?
It's
That it's made from corn, it's natural, and like sugar, it's fine in moderation.
love that top. Well, thanks.
Get the facts. You're in for a sweet surprise.
So what we're noticing here is, I think it's funny that they have the lady kind of looking, she's blank-faced about why high fructose corn syrup's not good. Again, think about why it's not good. It's, number one, it's not natural. Number two, it's a highly concentrated sugar that you're exposing your body to. There is studies that show that, rats that are constantly exposed to high fructose corn syrup do become obese. I just wanted to show you this one just because I think it's funny. Saturday Night Live did a skit too.
The following is a message from the corn syrup producers of America.
Wow, you don't care what the kids eat, huh?
Excuse me?
That has high fructose corn syrup in it.
And?
Well, you know the things they say about high fructose corn syrup.
Like what?
Well,
That it's made from corn, it's natural enough, and like sugar, it's fine in moderation?
I guess...
You guess what? That you should have kept your mouth shut?
I never...
Never what? Never heard of science? This is a real jam for me. Trust scientists or stay-at-home mom Sheila from down the street who's having wine at 10 a.m.
I'm sorry, let's just...
Let's just what? Make a bigger deal about the corn syrup? At this fun party that I've invited you to, even though I didn't want to because you say like this?
Okay. Hey, that's a great sweater.
Thanks. It was my daughter's, but she grew out of it.
Mommy! Can I have some more juicy drink?
Of course you can.
Yeah!
She's cute.
Yep.
Get the facts. Check out our website.
So I thought that's cute. One last thing. I think it's interesting to think about things in the big picture. Sugar and the roles of sugar. So again, like I said before, you do need sugar in your diet. But how your brain receives sugar is something that's being looked at further. Think about this, for example. No one's ever gotten morbidly obese eating just a fruit diet with just, the only sugar source they're getting is a natural source of fruit. What we're eating is processed food with this processed high fructose corn syrup. So an interesting documentary. This is on Netflix. It's really, really, really good. I'm just going to show you guys the trailer, and this is the last thing I'm going to say about high fructose corn syrup. I just kind of want to get you guys thinking about it, like what's in how, just kind of think about, this this topic.
The epidemic here is worse than previously estimated, much worse.
The message has been pushed on us. It's your fault you're fat.
Shouldn't be so hard to get them to run around and play, right?
They have voracious appetites and they don't exercise enough.
It's about how active our kids are.
Forget about it.
There are 600,000 food items in America. 80% of them have added sugar.
Your brain lights up with sugar just like it does with cocaine or heroin. You're going to become an addict.
You end up with one of the great public health epidemics of our time. This is the first generation of American children expected to lead shorter lives than their parents.
I am 12 years old, and my doctors have said that I am a statistic.
We're blaming willpower, and it's a crime.
Over 95% of all Americans will be overweight or obese in 2 decades.
We're toast as a country.
The sugar industry is extraordinarily powerful. They're in business to make money, not to keep America healthy.
What if our whole approach to this epidemic has been dead wrong?
The government is subsidizing the obesity epidemic.
To place private profit ahead of public health. Systematic political failure.
By 2050, 1 out of every 3 Americans will have diabetes.
Those diseases are being driven by sugar.
This is the fundamental problem nobody's talking about in society.
We could cure 80% of the problem where they prepared the food in the school.
Tomato paste is a vegetable? Really?
Junk food companies are acting very much like tobacco companies did 30 years ago.
I would reject entirely any argument that they are in any way harmful.
Lying through their teeth.
Kids are being told the biggest lie they will ever hear in their lives.
Ronald McDonald never sells to children. He informs and inspires through magic and fun.
If a foreign nation were doing that to our children, we would defend our families.
Years from now, we're going to say, I can't believe we let them get away with that.
You have to change the diet of America.
It's all preventable.
All right, so just to kind of give you guys a summary of what you're would see in this video. Again, I highly recommend watching it. What this is kind of showing you is, let me add a little thing here. Let me bring my screen back up with a pen. Sugar. I think it's not working. There it is. So sugar is 8 times more addictive than cocaine. That means you would be more likely to give up a cocaine addiction than you would to give up sugar. Some things that the studies have also shown is that when you consume sugar, when your sugar levels get low, if you're hungry or not, it tells your brain that you need to eat again. It's one of those things where you cannot even be hungry, and because your body's low in sugar, that addiction tells you to go and consume something that has sugar in it just because you need it. It's like you're fiending for it, even if you're not, like I said, not hungry. So that could be the problem with obesity. You're eating when you're not hungry. You're eating because your body's going through withdrawals of the sugar. So that's kind of the whole idea of the Fed Up documentary. But high fructose corn syrup is a culprit here. It is a very concentrated, cheap sugar. That's the end of the high fructose corn syrup. All right, moving on to look at polysaccharides. Basically, if you want to think about a polysaccharide is essentially a carbohydrate. Normally, we like to call them starches because they're really large sugars. So let's think of it as a starch. But polysaccharide could also be, it depends. This is kind of where it gets a little dicey. Polysaccharides could be starch. A polysaccharide could be a carbohydrate. They're the same thing at the end of the day. Think about it. When you're making dinner, all right, you're probably gonna make some sort of starch, potato, rice, something, right? At the end of the day, potatoes, rice, they're all types of carbohydrates, right? So they're all the same thing. But one thing to kind of look for is that polysaccharides are essentially starches, but they're also a type of carbohydrate. Polysaccharides are macromolecules, polymers made up of thousands of monosaccharides. So remember, poly means many, many sugar versus made up of many one sugar molecules. Polysaccharides may function as storage molecules and structural compounds. These molecules are big, lots and lots and lots of bonds. So starch is a polysaccharide, all right? It's composed of glucose monomers and used by plants for energy storage. So these are coming from plants. And again, I listed them a minute ago, potatoes, rice, that sort of thing. Even if you think bread is a starch, right? But what's bread come from? Wheat. Well, a traditional form, wheat. Glycogen is a polysaccharide composed of glucose monomers and used by animals to store energy. Animals store sugar as glycogen. In the liver. So basically, any excess glucose that we have will be converted into glycogen and stored in the liver. With not all of it, but some of it. Cellulose is a polymer of glucose and informs the plant cell walls. Chitin is also a polysaccharide. Cellulose is also a polysaccharide. They don't have it in here. They should have written that. Cellulose is also a polysaccharide. It's an ingestible, non-digestible. So we cannot digest cellulose. And I think there's a picture coming in just a minute that I want to show you guys in regards to that. We'll look at it in just a minute. I have a story for this. Chitin is also a polysaccharide. It's used by insects and crustaceans. Think of things like crabs to build the exoskeleton. So this is going to be used for the exoskeleton of crabs and insects. And it's also found in the cell wall of fungus. So take a look at these. So this is starch, glycogen, cellulose. All right. So here's starch, and they're using a potato. And then they're using glycogen, which is stored in some muscle tissues. And then here's cellulose. Notice that they all look the same. Notice this starch and this glycogen looks almost identical. I mean, it is identical, right? We can digest both of these. These are things that our body recognizes and says, hey, you know, I know what to do with these. I can do it. I recognize this starch molecule. I can process it. Glycogen, our body can use it. We can break it down and use the sugar molecules. However, cellulose is different. Do you see how it's like there's a bond up, there's a bond down, there's a bond up, there's a bond down, there's a bond up. Here it's the bonds down, 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 down. Here. So again, it's down, up, down, up. That simple change in the bonding makes it where we cannot digest cellulose. Let me give you an example. Corn, okay? Corn, let me draw some corn. All right, so just imagine a corn kernel. All right, the outside is that capsule, right? That outside of the corn is the cellulose. What's inside of the corn, though, is starch. So we eat corn, most people use corn, maybe eat corn once a week or something like that. Here's the thing about corn. Let's say you cook a can of corn or a bag of frozen corn or something like that. You eat corn for dinner. You see the corn later. It doesn't just, you chew it, you eat it, and then you see it later. You can test your digestive system with corn. Here's the reason why. You don't completely process that corn because we can't process this cellulose. We can't break the cellulose down. So when you poop, there's the cellulose. That casing of the corn is not digested. The starch that was inside of the corn has been digested. The cellulose, the capsule of the corn, is still there. We can't process it. It's along the same lines as the fact that we don't eat paper because we can't digest it. So it shows you, just because they look structurally similar, structure follows function. We functionally cannot break down the cellulose. All right, polysaccharides are usually hydrophilic, means water-loving. Let's give an example of why that's important. Let's think about bath towels for a minute. Bath towels are made of cotton. Cotton, they're really good at drying you off and absorbing lots of water. The reason why is because they're mostly cellulose, therefore they're water absorbent. So take a look at this.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Moving on to our next macromolecule, and that's the lipids. Lipid. Let's think. What is a lipid? Lipids are fats, and how do you know that? I always kind of tell students to think about liposuction. Well, in the procedure for liposuction, you are sucking out what? Fats, right? So liposuction, lipolipid, lipid here, fats. So we're going to talk about the different types of fats. Lipids are water insoluble. They're hydrophobic, water-fearing compounds. These guys are nonpolar. So these guys are nonpolar. They are long-term energy storage molecules. These guys are huge, lots and lots and lots of bonds, okay? Contain twice as much energy as a polysaccharide. All right, is made up mainly of carbon and hydrogen, and again, nonpolar covalent bonds. These are not huge molecules when it comes to comparing them to carbohydrates. They're not built from monomers. Lipids do vary a great deal in structure and function. We're going to look at three types of lipids: fats, phospholipids, and steroids. A fat is a large lipid made from two kinds of smaller molecules, glycerol and fatty acids. Typically, it's going to be made up of a glycerol plus 3 fatty acids. All right. A fatty acid can link to glycerol by, again, you're putting these together by a dehydration reaction. All right. Fats contain 1 glycerol and 3 fatty acids. Fats are often called triglycerides. They're called triglycerides because you are going to need to make 3 bonds with glycerol, and I'll show you what that means. So here it is. So here's that 1 glycerol, and then there's the 3 fatty acids. So you're going to need to do 1, 2, 3 bonds, so that's why it's called a triglyceride. So let's take a look at this animation.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in 1 or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats, like butter and lard, are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
All right, so let's build on what he was just saying. Here's that glycerol with a fatty acid. Notice that you're doing here a dehydration synthesis. Here is another dehydration synthesis. This one is an unsaturated fat because of this double bond. Think about what this word means. Let's get back. Let me give you a little tidbit here. If this were, let's expand this out. So it's got the carbon, and I'm looking right here. All right, I'm going to draw it out. Actually, let's go back for just a second. I'm just going to clear this up just a little bit. Let's expand right here. All right, so that's a carbon and it's got 2 hydrogens. Carbon with a hydrogen and then a double bond and then a carbon with a hydrogen, and then we're going to go to another carbon. Okay, this is not saturated. Saturated means that you're holding all the possible things you can hold. Like you're holding as much as you can. You're saturated. You're completely consumed. This is not completely consumed. Why is it not? Because I could break this double bond here, put a single bond here, and add a hydrogen here and here. Now I am saturated because now all my bonds, all my carbons have 1, 2, 3, 4. 1, 2, 3, 4 bonds. I'm now saturated. I can have as many bonds as possible. Now you do have 4 bonds. Each carbon has 4 bonds here, but we're not holding as much as we could possibly hold. So that's why this is considered unsaturated versus this being saturated. Now, when you're looking at saturated, let's get back to my red. Saturated fats are going to be solid at room temperature. And unsaturated, this isn't fair that they have this fish here. Fish does have some unsaturated fats in it, but it also has saturated fats. So it's a little misleading, and I wish they didn't have it here because that's confusing. All right. Ignore the fish that's sitting here. The reason they put unsaturated fat here is because, especially something like salmon, it has some unsaturated fats in it, which is very, for an animal product, is very rare because animals can't make double bonds to make unsaturated fats. Some fish are able to make that unsaturated fat because of the temperature that they live in, so they're able to make the unsaturated fats. Typically, animals only make saturated fat, okay? Butter, what does it come from? It comes from cow's milk, right? A cow is an animal, therefore this is saturated fat. It doesn't matter if it's organic, okay? It doesn't matter. It's still unsaturated fat. It's just it's saturated fat. It's not good for you, all right? Saturated fat in the steak, it's saturated fat, okay? It comes from an animal. There's saturated fat in this fish here. It doesn't matter. There is unsaturated fat, but there's saturated as well. Saturated fat, unsaturated fats are typically only found in plants, okay? Animals cannot make that double bond that's needed for unsaturated fats. Again, I say fish is rare because of the temperature, but plants, if you're going to consume unsaturated fats, you have to get it from a plant source. All right, and we're going to expand on that as this continues. All right, so some fatty acids contain 1 or more double bonds forming unsaturated fats. Again, and we talked about that. Fats that have the maximum number of hydrogens are called saturated fats. All right, so unsaturated fats are referred to as oils. Most animal fats, so if it's coming from an animal, for the most part, it's a saturated fat. These are our bad fats again. And the reason why it's saying most animals is because there are a few fish, such as tuna and salmon, that because of their environment are able to make that double bond and produce unsaturated fats, polyunsaturated fats. Hydrogenated vegetable oils are unsaturated fats that have been converted to saturated fats by adding hydrogen. Some examples of this, especially in, let's say, like the 1950s, 1960s, was when they started using hydrogenation to produce margarine. Margarine is partially hydrogenated vegetable oil. This hydrogenation creates trans fats, which pose lots of health risk, especially cardiovascular disease. So let's take a look at this. Now, when we're looking at this, I'm going to give you an example. And we're going to look at peanut butter. All right, so when we're looking at peanut butter, let me get a pen here. So before we go there, let's again look at saturated versus unsaturated. We know saturated means I have as many hydrogens as I can have. So we said saturated, no double bonds. And we said animals cannot make double bonds. And then unsaturated has the double bond, right? So let's take a look at this. So let's say you're shopping for peanut butter. You're on the aisle and you're looking. Let's say you have that, I'm just going to give, just because Jif's on my mind. So you have that Jif peanut butter. And there it sits in this little jar. And it's all one consistency. It's nice and creamy and pretty. Okay, it's pretty. There it looks, all creamy and nice. Then let's say you have this all natural peanut butter or something. And just for the sake of being clear, just because it says all natural doesn't mean it isn't good for you. All natural something other peanut butter. And maybe it looks like this. It has this jar, but on the top of the jar, there's these oil. Like there's this oil on the top. This is an ugly drawing, but it'll just have to work. So there's my, so it still has this nice consistency, but all of this, let me just, here's my, so this layer right here, I'm going to draw in blue. Actually, let's do it in yellow. All of this yellow is liquidy. Liquidy oil. Okay. Which one's good for you? The pretty one or the not so pretty one? Most people would think, oh, well, this one's old. You know, this is an old peanut butter. Why does it look like that? You know, it's old. But the truth is, this guy here is good for you. It's the better one. What Jif and other nut butter companies have done is they've hydrogenated. So this right here, this liquid would be your unsaturated fats. And this is going to be your saturated fats. So all this is going to have saturated fats. What they did is they've made all of this saturated. I think they do leave some unsaturated fats to be completely 100% with this. There are still some unsaturated fats, but they have hydrogenated so much that it allows it to keep this consistency. But it's called hydrogenated. They have taken, so this yellow here would have bonds that look like this. You know, the double bonds. When you hydrogenate it, you break that bond and you saturate it with as many hydrogens as you can. When you do that, it's not natural, it's not healthy. That is what leads to trans fat. It's not a natural fat. It's this, you know, unnatural fat. So something that's not a naturally existing fat. Something that's artificially made, your body can't recognize, and it's not good for it. All right, so that's the deal with the peanut butters. So now we want to look at, so this is just kind of explaining this little scientific thinking section, just looking at the effects of trans fat. At the end of the day, trans fat is like a saturated fat, but it's like a saturated fat on steroids, if I can make an analogy here. The problem is hydrogenated oils. Think about something like Crisco, like something that lard. My grandmother used to cook biscuits with lard in them. I'm from the South, so to me it was nothing to eat a larded biscuit. They literally, it was like a vat of like fat that they would cook, that people would cook with. It was natural. I think that's considered a type of saturated fat, but I think some of like the Criscos and things like that are completely hydrogenated. But these are like animal fats. And then they take the vegetable oils and they hydrogenate them to make things like Crisco. So in the 1890s, the process of adding hydrogen atoms to unsaturated oils was developed. It helped reduce spoilage of oils. It helped oils withstand repeated heating for frying. So to be able to keep your grease around, to be able to keep using it. Reduce consumption of animal fats, thought to be less healthy than partially hydrogenated vegetable oils. By the 1990s, partially hydrogenated oils were common in cookies, crackers, snacks, baked goods, and fried foods. So now they have to label them, and when it comes to these trans fats, in 2006, the U.S. Food and Drug Administration required the listing of trans fat in food labels. Here's the thing, though. They require these companies to list the trans fat. But do you know that they don't use very good with the USDA? Companies can list something that has 0.5 grams of trans fat as 0. So something can say trans fat, 0 grams, but in fact, it has 0.5 grams in it. Think about the magnitude of that. Let's say you have something that's got a serving of 32 grams, and it says trans fat 0. If you eat 64 grams of it, you've now had 1 gram of trans fat. That's a lot of trans fat that's hidden. It's hidden trans fat. The thing is, they allow people to get away with it. That, to me, this should round up to 1. That's 1 gram of trans fat. Any math teacher will tell you that 0.5 is going to round up to 1. But for some reason, the FDA allows these companies to round that down to 0. Doesn't make any sense. So there's studies for this. There's a lot of countries that have completely banned hydrogenation and banned trans fats. We are not one of those countries. So what this is showing is it's hard to do an ethical study of this, especially using humans, because you don't want to prolong people's exposures to trans fats because it's someone's life. I mean, you have things like, if you want to think of, what is that movie called? The guy who ate McDonald's for like 30 days or something like that. What is that? It'll come to me in a minute. I can't remember the name of the movie, but it's not. They don't want to constantly expose people to this. There was a study that was done. Obviously, what you want to do, they did observational scientific study on dietary health effects, extended over a long period of time, used more representation of population, and measured disease outcome and risk factors. The nurse health study began in 1976 with more than 120,000 female nurses, and it is the example of an observational study. They didn't actually tell these nurses what to eat. They just said, hey, we're going to just observe your life. Let's see. So what we have here is this risk table. So I think they ended up using, as far as studying the nurses' fat, the dietary fat, they used 80,082 women from 1980 to 1994. And here's the table. So 1 represents the risk factor. So what this table actually shows, let me get my pen, is that if 1 means that there is no difference in the risk, if these women, Saturated fats increase their risk of heart disease by 17%. To consume monounsaturated and polyunsaturated fats decreased their risk by 19%. And polyunsaturated decreased their risk by 38%. By consuming trans fat, they increase their risk of heart disease by 93%. That is substantial. All right. Super Size Me. I was sitting here like, what's the name of the movie? So the movie I was referring to earlier, it just kind of came to me. Super Size Me is probably, if you are interested in like how, again, he only did the study for 30 days and he only ate McDonald's, which McDonald's is notorious for having their trans fat. It's in most of the burgers, I think, but he just ate McDonald's. And so that was an example, but that was something that he chose to do. I think ethically these people didn't want to expose people to long periods of time. So, but Super Size Me, that's the name of the so if you've seen that, that'll kind of give you an idea of exposing yourself for 30 days to trans fat. All right, so why do we need unsaturated fats anyways? Like, so we're looking at saturated fats versus unsaturated fats. Which one is good, which one's bad? We said already that saturated is bad and unsaturated is good. Well, why are they good? That's because we can't make unsaturated fats. We need them and we can't make them. So let's go ahead and write that down just to be clear. So unsaturated fats. We need them. We cannot make them. All right. Why do we need them? Why do we need them? That takes us right to phospholipids. Phospholipids are the major component of all cell membranes. Phospholipids are structurally similar to fats. Fats contain 3 fatty acids attached to a glycerol. Phospholipids contain 2 fatty acids attached to a glycerol. This is a phospholipid. It's called a phospholipid because it has that functional group that we discussed, the phosphate group here. So, my mouse, there it is. So this is a phosphate group here. I like to abbreviate this like this. We always kind of draw it and then something like that, or that's kind of like that's ugly. Both of them are ugly, but this one's better. This is how I typically draw the membranes. So when you're looking at a membrane, so when we talk about cells, so if this is a cell, right? The cell membrane is made up of this. So if I were to blow this little section up and look at it, it would look like this. It's this double phospholipid membrane. This is the top part, the head region, and then these are the tails. These are the phospholipid tails. Notice this has a kink. The kink is from that unsaturated fat. All right. Every cell in your body it's got these phospholipid membrane. That's why you need the unsaturated fats. You need to be able to build these membranes. You can't do that without consuming unsaturated fats. This is the phospholipid. The top of it has all these negative charges. The top part here is polar. Remember, water's charged, right? So this part, let's go back. Let's change right. Come on, pen. There we go. This part here is polar. So it's hydrophilic, water-loving. And all of this, nonpolar, and then water-fearing. Think about why that's important. if this is your cell membrane right here, this guy here needs to be nonpolar. You don't want water to come through and just collect in the middle of your membrane, because that could cause the cells to bust. So this part here, the outside is water-loving, this is water-fearing. Phospholipids cluster in a bilayer of phospholipids. The hydrophilic heads are in contact with the water in the environment and the internal part of the cell. The hydrophobic tails cluster together in the center of the bilayer. So that's this picture here. So again, this is called a phospholipid bilayer because you have two layers. All right, so steroids are lipids, in which the carbon skeleton contains 4 fused rings. We looked at that earlier. We looked at steroid and we looked at the estradiol. Cholesterol is also a common component of animal cell membranes and a starting material for making steroids, including your sex hormones. So cholesterol is used to help make steroids. Now, cholesterol always gets a bad rap. Normally when you hear someone discuss cholesterol, it's because probably they have cholesterol issues. Maybe they have high cholesterol and that sort of thing. So another thing, studies have kind of shown that, unsaturated fats are important in keeping the bad cholesterol low. So that, is something else to kind of think about. It's a whole other thing. But when we're looking at cholesterol gets a bad rep because most often people assume that cholesterol is all bad. Well, cholesterol has a lot of good roles in the body. And again, with helping produce steroids, but also with producing vitamin D. Cholesterol and the UV light. So let's say you go outside and you're exposing yourself to UV from the sun. Cholesterol and the UV produce vitamin D. So this is something where cholesterol is useful for the body. This is cholesterol here. So if we look at anabolic steroids, this is are synthetic variants of the male hormone testosterone. These are what they're used by athletes, bodybuilders, things like that. So there's always been that, you may have heard roid rage or something like that. Anabolic steroids are not their synthetic version of testosterone. There's a lot that could happen from your body. Violent mood swings, depression, liver damage, cancer, high cholesterol, high blood pressure. A man could become sterile because here's what things, your body works on something called a negative feedback. And so when you're introducing all this anabolic steroids, all this testosterone, your body's like, well, you've got enough testosterone. It's going to stop making testosterone. So the only source of testosterone you're going to get is from your anabolic steroids until you stop taking it. Once you stop taking it, then your body will go back to producing testosterone. But as long as you're taking anabolic steroids, your actual natural steroids will probably cease to be made. If you're not producing testosterone like you're supposed to, then you're not going to produce semen. And so your sperm count is going to be low. It could cause you to become sterile. So anabolic steroids, liver damage is a big thing, cancers, so all these other things, mood swings. All right, proteins. Proteins are involved in nearly every dynamic function in your body. They're extremely diverse, with tens of thousands of different proteins, each with a specific structure and function in the human body. Proteins are composed of different arrangements of the common set of 20 amino acid monomers. Most things in your body, everything about you, from the hair color, your skin color, your eye color, everything is essentially protein. It's genes that code for protein, so ultimately it's like a protein expression. Okay, your skin color, the amount of melanin that your body's making. What is melanin? It's a type of protein. Everything is protein, 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 protein. Enzymes, proteins. Most things are proteins. If you have to guess on a test, the answer is protein. Most things are proteins. Probably the most important role for proteins is enzymes, proteins that serve as catalysts. Catalysts are going to Proteins serve as catalysts. Catalysts speed up reactions. Regulate virtually all chemical reactions within cells. You're not going to be able to break, you can eat food all day, but you can't break it down unless you have proteins. So they're involved in all your chemical processes. You have transport proteins, you have defense proteins like antibodies in the immune system, you have signaling proteins in hormones, chemical messengers. I mean, proteins are everywhere. Most people, when they think of protein, just think of meat. Okay, everything has protein in it. Protein is everywhere. Other types of protein include receptor proteins built into cell membranes, contractile proteins found in your muscle cells. Muscle cells only do one thing, contract. That's it. Well, they two things typically. They contract and relax and contract and relax. Contractile proteins within that muscle. Structural proteins like collagen. You have storage proteins, which serve as a source of amino acids for developing embryo and eggs and seeds. The function of different types of protein depend on their individual shape. A polypeptide chain contains hundreds or thousands of amino acids linked together into peptide bonds. Amino acid sequence causes the polypeptide to assume a particular shape. So this is a protein. This is the same protein but a different, a different model. This is protein for spiderweb. So spiderwebs, the protein that they spin is just phenomenal. There's a lot of research that goes into these spiderwebs. If a protein shape is altered, it can no longer function. That process is called denaturing. So if you're denaturing a protein, you unravel it and the protein will lose its shape. Remember, structure follows function. If it changes its structure, it's changing its function. Proteins can be denatured by changes in salt concentration, changes in pH, or to heat. You can freeze, most often you can freeze a protein and it doesn't alter their shape. But once you heat it, you destroy it, essentially, you denature it. The monomer of a protein is an amino acid. All right, so this is the monomer. Or you could say the building blocks. For protein is amino acids. The amino acid is made up of, remember those functional groups we talked about? An amino group and a carboxyl group, which makes up for that whole acid. It's actually called a carboxylic acid. And also bonded to a centrum atom with a hydrogen atom and a chemical group represented by R, which determines the specific properties of each of the 20 amino acid proteins. So let me show you what I mean. This is an amino acid right here, just a very generic amino acid. So this is a generic amino acid. Everything is going to stay the same. There's 20 different types of amino acids. The only thing that changes is the R. This is what changes. You have, this is an amine group, and this is our carboxyl group. All right. An amino acids are either classified as hydrophobic or hydrophilic. It just depends on what that R group is to determine if it's hydrophobic or hydrophilic. So leucine would be an example of a hydrophobic because this guy here, this would be a hydrophobic condition because these are all covalently bonded. This, we know oxygen is very greedy, This is going to be a polar. This guy's polar, and this is polar. My A's look like O's here. Oh, I'm sorry. Polar, not whatever that looked like. So here these are both polar, so therefore they're hydrophilic. Okay. Remember, these oxygens don't share well, right? Oxygen's not a good sharer, so it's not going to share electrons. If it's not sharing, it's going to be polar. Amino acid monomers are linked together through dehydration synthesis, just like everything else. The carboxylic group of one amino acid is bonded to the amino acid of another group. That's going to create something called a peptide bond. Amino acids can be added to in the same process to make an amino acid chain. So just to kind of clarify, let me see what picture we have next before I write that. Go. Okay, so here's that process. And let me get my pen. All right, so you have an amino acid plus an amino acid. Whenever you want to bring these two amino acids together, you're going to do a dehydration reaction, and you're going to form a peptide bond. If let's say you put 10 amino acids together, you've made a polypeptide. That just means many peptides. Many peptides. You will often hear a protein called a polypeptide. Sometimes they're used interchangeably. At the end of the day, a protein is a polypeptide bond because you're going to make a protein by bonding together lots of amino acids. Right. So at this point, I've kind of misled you a little bit. I said that a protein is essentially a polypeptide. It is, but it isn't. And let me just tell you why. It's not quite true. The distinction between the two is analogous to the relationship between a long strand of yarn and a sweater of a particular size and shape that you could knit from the yarn. So what I kind of mean from that is the functional protein is not just a polypeptide chain, but it's one or more polypeptide chains precisely twisted, folded, or coiled into molecules of a unique shape. We can kind of dissect the overall shape of a protein. You have a primary, secondary, tertiary, and quaternary structure. Proteins with more than one polypeptide chain have a fourth level quaternary structure, and we're going to kind of take a look at those. Now, there's some videos. I'm not really going to go through and have you guys sit with me to watch the videos, but we'll just kind of look at a couple of them, maybe the first introductory ones.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
I want to just elaborate a little bit here. The reason why it's important that you kind of understand what I'm saying here. This little bond here, let's get this away. So they were showing you here, this little bond, my pen is not wanting to come up. There it is. They showed this as being a peptide bond, right? So every one where there's a connection between it looks like little beads. These are all peptides. So all of this would be a polypeptide chain. And this is a protein, right? It may not be a functional protein, but it is essentially a protein. Each ball here is an amino acid, so that's why it's considered the monomer. So let's just reiterate, monomer is the amino acid, and the polymer is the protein, or the polypeptide in this particular situation. Now, so we looked at this one already.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
So looking at the tertiary protein structure.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
And then the last one's, this is the last one.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
So just kind of looking through here, here's the idea. In this linear form here, you're looking at the primary structure. When it starts getting characteristics like an alpha helix or beta sheets, that's the secondary aspect. When you have interactions between, so you're looking at interactions between those R groups, then that's your tertiary structure. The quaternary structure isn't always present, but it's when you have multiple polypeptides, polypeptide chains that come together to make a functional protein. So that's the gist here. All right, our last macromolecule is nucleic acids. And you may be thinking, what in the world is a nucleic acid? Well, what we're talking about here is DNA deoxyribonucleic acid and RNA. It is the DNA that's going to come up with the amino acid sequence for the polypeptide programmed for all of our, we are who we are because of our protein expression. Our protein expression comes from the genes. Genes consist of DNA, deoxyribonucleic acid, which is one of the types of nucleic acids. We know that DNA is inherited from an organism's parent. DNA provides directions for its own replication. DNA programs the cell's activity by directing the synthesis of proteins. DNA does not build protein directly. DNA works through an intermediary molecule called RNA, which is ribonucleic acid. DNA is transcribed into RNA. That's an important word here. which we'll come back and look at in later chapters, but this word transcribed. So DNA to RNA is called transcription. And then RNA is translated into a protein, and that's called translation. This is going to happen in the nucleus, and this happens in the cytoplasm of cells. And at this point, you do not know what a cytoplasm is. Let me just show you real quick. This is a cell, right? This could be the nucleus. This stuff here, all of this is cytoplasm. Now you know what cytoplasm is. This is DNA. DNA is always double-stranded. Just like you're seeing here. Translated into RNA. RNA is single-stranded. And then RNA is translated into proteins. DNA's monomer is nucleotides. Okay, so I want you to lock this in right here. I'm going to draw you a picture. Don't forget it. So monomer of DNA is nucleotides. The monomer of RNA is nucleotides. So the monomer of nucleic acid is nucleotides. Nucleotides have three parts: a 5-carbon sugar called a ribose in RNA, and deoxyribose in DNA. They also have a phosphate group and nitrogenous base. How do we remember that? Everybody think about suburban America. What's in suburban America? You have, everybody wants a house, right? A pool, and maybe a garage or some sort of fancy driveway. There it is. So we have 1, 2, 3, 4, 5. There's my 5-carbon sugar. This is my phosphate group or my pool. And then this is my nitrogenous base. So think of this as a house, a pool, and a driveway, or a garage or something. This is a nucleotide. What's going to change here is the sugar. If it is an RNA, it's a ribose sugar. If it's deoxyribose, if it's DNA, it's deoxyribose. It's still a 5-carbon sugar regardless. This is a phosphate group and sugar and a nitrogenous base. So it's your sugar, your phosphate, and your nitrogenous base. So this is a nucleotide. DNA is going to have 4 possible nitrogenous bases: adenine, thymine, cytosine, guanine. RNA also has all of these except thymine. RNA does not have T, it has U. All right, these guys pair up very specifically, and I'm going to try to write it here. So when these guys pair up, A is going to pair with T, C is going to pair with G. In RNA, A is going to pair with U, and C will still pair with G. Okay? All right, a nucleic acid polymer, or a polynucleotide, forms from a nucleotide monomer when the phosphate of one nucleotide bonds with the sugar of the next nucleotide by dehydration synthesis. This produces a repeating sugar phosphate backbone with protruding nitrogenous bases. So let's show you what this looks like. So this is just kind of showing you a sugar phosphate backbone, and then this is the nitrogenous base. Not the best picture. Let's see if there's another one. We'll see another one coming up in just a second, and I'll go through what that means. RNA is usually single-stranded, Where DNA is double-stranded, it's got that double helix. Two strands are associated because particular base pairs of hydrogen bonding with one another. And like I said before, A pairs with T, C pairs with G, producing base pairs. So C and G, T and A, C and G. So we know this is DNA because it's double-stranded and it has the letter T in it. Let me just kind of draw something here to make this clear because I don't think this picture is 100% clear. Remember when I was showing you, let me go back. My pen's not working. Select it again. Nope, that's not the one I want. This one. All right, so here's my nucleotide, right? And let's say, I'm just going to draw T, how they have it drawn here. Like this. It's an ugly one. All right, what happens here? Let's put another one up here. You form a bond here. And then this one would, there's one up here. There's a bond that forms here. And so forth and so on. This is that sugar phosphate backbone that they're talking about. That's what's represented here. All right, so by looking here, I want to kind of give you a little review as far as like what to kind of take from this. When we're looking at, we looked at the 4 macromolecules. And so when we're looking at, let's say, monomers, we could say monosaccharide. The polymer here could be polysaccharide. Sometimes we say glucose is a monomer, and we could just say carbohydrate. You could say glucose, and your polymer could be starch. All right, you're really, just to kind of give you an idea. When we're looking at amino acids, amino acid is a monomer. The polymer here is protein. Or you could say amino acid, or you could also call it a polypeptide. It's the same. And then when we say nucleic acid, oh, that's not what I want. I don't know how to erase. Nucleotide would be the monomer, and the polymer would be nucleic acid. You could say DNA or RNA. Notice I don't have one here for lipids. Lipids don't really have monomers, so it wouldn't fall into this category. This is in the back of your book. I would recommend printing this one off. This is really, really great as far as looking at the different macromolecules with their function and giving you some examples. I would highly recommend going through this. All right, if you have questions, make sure you leave it in the comments below or send me an email and we'll go through it. Okay, that's it for chapter 3.